Hello everyone, welcome to Get My Class. Let's solve this question which says annealing occurs at temperature of option A 55 degrees Celsius, option B 74 degrees Celsius, option C 98 degrees Celsius, and option D 40 degrees Celsius. So let us understand the question. The question says annealing occurs at the temperature of. So they are asking at which temperature annealing will occur. So first of all, we should know what is annealing. So annealing is nothing but binding of primers. Okay, this is seen especially during PCR, that is polymerase chain reaction. We see this process which is called as annealing. So what is usually annealing? Annealing is nothing but binding of primers, or you can say oligos. It can be, you can also call it as oligos. Okay. Binding of primers to the single stranded region of DNA. Single stranded DNA, or region of DNA, because sometimes what happens, the entire stretch will not be single strand, it will be double strand, but then in between part of it will be in the form of single strand. So anyhow, the binding of the primers will occur to the, it will happen only at the single stranded region of the DNA. Okay, that is what is annealing. Okay, what is annealing? That is binding of the primers or oligos to the single stranded region of DNA. Okay, now there is a particular temperature at which the binding occurs. So it is around, you can say, it is around 45 to 60 degrees Celsius. Okay, so in between that, whenever the temperature is there, in between that, annealing will occur. Okay, so during PCR, that is polymerase chain reaction, you can see that the primers will bind to the single stranded region, between the temperature, when the temperature is 45 to 60 degrees Celsius. Okay, so let us see. Option A says 55. So yes, we can consider this because it falls between 45 to 60. So this is the correct temperature at which annealing can occur. Then 74 at this temperature, like 72 to 74, you can say, there will be extension which will occur. Extension in the sense, during polymerase, you can say polymerase chain reaction, the TAT polymerase, which is thermostable, that will bring about extension. That means it will add polymer of, it will create a polymer of nucleotides. So it will extend by addition of nucleotides. Okay, so this is extension. Hence it is ruled out. So at 74 degrees Celsius, it will not get annealed. Okay, so that we have to keep in mind. Now option C says 98 degrees Celsius. So at 98 degrees Celsius, like 94, 95 to 98, it will be denatured. The double-stranded DNA will get denatured to open up. It will open up and it will form single-stranded DNA. That is called as denaturation. So 98 degrees Celsius, it will cause denaturation, okay, of the DNA. Hence this is ruled out. Then option D says 40 degrees Celsius. So at 40 degrees Celsius, what can happen? Most probably the double stranded DNA again it will renature and it will become like the single stranded DNA again it can undergo renaturation and it will form double stranded DNA. Okay, and hence it is ruled out. So at the annealing will occur when partially there will be single stranded region in the DNA, and that happens at the temperature which is denoted by Tn. So, but here specifically we are talking about annealing where the primers will bind to the DNA. So the correct option is option A, that is 55 degrees Celsius. So let us conclude the answer. That is option A. I hope you understood the explanation. Thank you everyone.